Welcome to today's webinar, which is being offered as part of the Energy Training for Ag Professionals project. My name is Mike Morris. I work at the National Center for Appropriate Technology, and I'm one of the co-chairs of the Energy Training for Agriculture Professionals project, along with Steve Moore of the Center for Environmental Farming Systems. Funding for our project and for this webinar come from the southern region of SARE, which is the Sustainable Agriculture Research and Education Program of the U.S. Department of Agriculture. Our speaker today is Dr. Gary Hawkins. Gary is an agriculture pollution prevention specialist in the Department of Biological and Agricultural Engineering at the University of Georgia. Gary will be speaking to us today on the topic Onion Power, Potential Production of Biofuels from Onions and Other Vegetables. We strongly encourage you to ask questions during the presentation. Just raise your hand by clicking the small hand icon toward the upper right-hand corner of your screen, and either Gary or I will unmute you so everyone can hear your question. And with that, I will turn it over to Gary.
Thanks, Mike, and welcome everybody. Mike asked me to do this presentation on some of the anaerobic digestion work we're doing, and specifically looking at some of the onion work that we've been doing, and then in combination with the onion work, we've been doing some vegetable work. So we'll go ahead and get started. Like Mike said, I do work for the University of Georgia in the Biological and Ag Engineering Department, and I'm actually located in one of our off-campus sites in Tifton, Georgia. And I've got started in this field of anaerobic digestion, specifically with fruit and vegetable waste, particularly. I started off here at UGA in water quality and looking at erosion, runoff, water quality, and how all that's affected. And vegetable industry here in South Georgia is one of the major industries. So we started working with that group and noticed that a lot of the materials that they actually harvest in the field, a lot of that material, and I'll show you some of those numbers in a minute, but a lot of that material goes out the door to grocery stores and whatnot, but a good portion of that, or some portion of that's culled out. And so my background in graduate school, at least, was dealing with anaerobic digestion. So this project and some related projects came out of trying to relate how do we deal with a waste product from this production industry, and then also how do we get some energy out of it and also protect water quality. So we'll go ahead and get started. This is one of the little cartoons I found a couple years ago, or I guess a couple months or so ago, especially in the animal husbandry and the animal waste management area. We always look at food going into a cow's mouth and then manure coming out the back, or if you want to think of it as an energy product, especially as we go through anaerobic digestion systems. So, you know, I started looking at, well, if we can use animal manure, where the cow uses some of that energy for its processes, why couldn't we use the feedstock that they're eating, in this case fruit and vegetable waste, to actually produce that energy? So that's kind of where the idea came from. Now, why use vegetables, especially here in Georgia? One of the reasons is, as you see there on the screen, roughly 860 million pounds of fruits and vegetables are produced annually and shipped out of the packing houses on truck to grocery stores and supermarkets and wherever else annually. I think last year it was pushing a little bit higher than that, maybe in the billions of pounds, 1 billion pounds or so. But anyway, it kind of floats up and down around that number year in and year out. The second bullet there, as you see, some farmers and some packing houses actually during off season for vegetables here in Georgia will grow fruits and vegetables overseas and then actually import those to Georgia or grow them in different parts of the country. For instance, one group here may grow onions in Colorado or Utah or New York and import them during the off season for Vidalia onions in Georgia. And so they have to bring those onions in, they have to repack them, and in that repacking process, there's a lot of culls. And then some estimates from some of the managers of these packing houses suggest that there's roughly 5 to 8% of that harvested fruit and vegetables actually goes out the back of the packing house, if you want to call it that, in form of culls. So that may be if the order for that day is for large cucumbers, if the workers pick medium and small cucumbers, there may be nothing wrong with those cucumbers, but that doesn't fit that day's order. So those small and medium cucumbers go out the back, or if there's a blemish on the fruit or something like that, a lot of that material will go to the cull piles, and that's the fruit and vegetables that we're looking at using. And another thing, this 860 million pounds of fruits and vegetables that we actually harvest annually and actually sell, there's probably that much, if not even more, of this material left in the field that is even unharvested. This is just a picture of just one day's, and not really one day's, but one test plot of vegetables that we pick here on Tifton campus. The main thing you see there is eggplants, those little brown shriveled up things, about 2 or 3 day old eggplant. You see some tomatoes in the background. Here, a couple pumpkins and whatnot. But if this is what we get off of our just small research plots here, you can imagine the fruit and vegetable waste that we could potentially get off of hundreds and hundreds of acres in production scale agriculture. So when we start looking at these culls, waste disposal has to come into the mix. And like any industry in America or around the world, waste disposal is a necessity, and it's a necessary part of that business. And in the business we're looking at here is fruit and vegetable waste, or fruit and vegetable production. So waste disposal is going to be a major part of how do we get rid of these things. And so one of the companies I've been working with is an onion production company. During Vidalia onion season, they have said they get close to 300,000 pounds of onions annually, not annually, weekly, that they cull out at their packing house. And that may be weekly for a couple months. During the off season, they may drop that down to half or third, but still, they're looking at huge numbers of potential waste materials that have to be dealt with there in their fields. Excuse me. But some of the problems we have with this waste material, as you would expect, if you've ever left a tomato, a bell pepper, a cucumber, anything on your kitchen counter and forgot about it for a day or so, as that material rots or goes through its processes, these fruits and vegetables are roughly 85 to 95% water. So as they start to decay, that water is released, or that liquid's released along with all those sugars. And so you have this, eventually you have this liquid form of material. Again, they're 85 to 95% moisture. And in that moisture, you have a lot of these sugars that we have to deal with as a waste material. This waste is also high strength, and when I say high strength, I'm talking about as it's measured by this COD, as you see here on the screen. And COD is chemical oxygen demand. And chemical oxygen demand is a general test that we can run on wastewater materials. It's a measure of how much oxygen is required for those chemicals within the test to break down all the carbon-containing compounds and reduce it down to carbon dioxide and water. So it's just a process. It's a chemical test that we can do to give how strong that wastewater is. And then from chemistry and stoichiometry, we can look at, for every gram of COD, we reduce or take out in an anaerobic digester, we can get 1/3 of a liter of gas of methane output from that. And a little bit later on in the presentation, I'll show you some of the COD numbers from the fruit and vegetable waste that we've been analyzing. And then also it can be a potential water quality problem. Like I said, when I first started here at UGA, I was mainly looking at water quality issues, working with this fruit and vegetable guys as well. Just the thought of them putting their materials, and a lot of these guys will spread them back on the field on their production fields and plow it in. Well, if they don't get to that material in time to plow it in and it becomes a liquid, it potentially could become a water quality problem, either through base flow through the groundwater or surface water. So what's the solution that we're looking at? Mainly, we want to look at managing this waste material, not only as a waste material, but looking at it as a feedstock for the anaerobic digestion process, and then hence getting that methane, getting that energy output from this fruit and vegetable waste that took energy from the solar, bounded up in this fruit and vegetable. We want to try to capture some of that energy out and then use it either in the packing facility, put it back on the grid if there's enough there, or use it in some other form or fashion. And then also the effluent water and the sludge that comes out of these anaerobic digesters can be used as a, if you want to call it, a low-grade fertilizer. Nitrogen, phosphorus go right through the system. They're generally not consumed in the anaerobic digestion process. So the effluent water, the water that comes out of the digester after reaction has occurred, and the sludge from the bacterial mass and whatnot can be good soil amendments. To show this a little bit better, I borrowed this slide from Dr. Ann Wilkie at University of Florida at IFAS there. But as you can see, it's pretty much the whole biogas cycle, as she's titled it. You've got your solar energy that goes into the field crops. That's either to grow the tomatoes, the bell peppers, the cucumbers, whatever. Those energy crops, or those crops, can then go into feeding animals. They can be used for biofuel production, whatever that may be. It could be diesel or some other biofuel production. Obviously, we can harvest it. Humans consume some of it. It goes into industrial processes to make ketchup or what other products, and then the waste material from that. But anyway, however it goes, it all eventually can end up as an organic waste. Well, then we can take that organic waste, either combine it or take the pure energy crops, as you see, and put them in anaerobic digesters. And these anaerobic digesters are vessels, various sizes, from small vessels of the ones I'll show you in a little bit are 90 liters up to millions of gallons. But these anaerobic digesters are closed systems. There's no oxygen entrained in them, and we try to keep as much oxygen out of the system as possible. And in that process, like I'll show you here in a couple of slides, the bacteria will take the organic waste or organic materials, break them down into that carbon dioxide and methane. Generally speaking, if you've got a well-run operation and everything's clicking along just like it should, you'll get somewhere between about 70-75% methane in your gas, and the rest, the 25-30%, will be carbon dioxide. And then a little bit of that carbon dioxide or methane percentage will be hydrogen sulfide and some other gases, depending on what the feedstock is. As you see with the orange line there, then the biogas itself can be done as the way Dr. Wilkie shows, is two different outputs. You can have electricity production where you run that biogas through a modified diesel engine or a modified engine, and then it's just a generator produces electricity for either on-site use or it can go back out into the grid. Some farmers in Vermont and some other places have actually gone through and cleaned the gas, got a lot of the sulfur and any other compounds in there, gotten them out of the gas, and you can, working with the pipeline, the gas pipeline, can actually put that back on the pipeline. One thing we're looking at is, and probably the most efficient way of using that gas, if there's an outlet for it, is to convert that gas into heat. So just basically burn the gas, produce either steam for a steam operation if you can do that, and you have the facilities to use steam, or just produce heat. And if you can use the heat there available, you'll have the heat there available. Then the other part of the anaerobic digestion process is that effluent water or the sludge. And as you see there with the green line, you can then put that back on the field as a biofertilizer. It's not necessarily a super fertilizer, but I guess more of a low grade fertilizer because, like I said, the nitrogen and the phosphorus really pretty much just flow right through the system. So the nitrogen and the phosphorus are located in that effluent water and the sludge, so that can go right back out on the field to produce more crops to continue that cycle. Excuse me. So some of the advantages of these anaerobic digestion processes, one is the process is real stable. If you are feeding these digesters a constant COD, and again, like I said, I'll show you some COD numbers here in a little bit, a constant COD diet, if you want to call it that, to the digester day in and day out. And once the bugs, the bacteria within the digester get used to what's going on, what they're eating, they'll produce methane on a constant basis. They'll produce it day in, day out, 24/7, as long as you're feeding the material. There's some basic monitoring you might want to do on it, and you have to do, just kind of make sure the process is working right, but generally it'll be a stable process. The output from these digesters is methane gas, and that methane gas is then a very available energy source, again, like we talked about in the previous slide, to be used in many different ways. A lot of animal operations would use these anaerobic digestion systems as odor reduction methods or odor reduction facilities. Some of them may not want the methane output, and they may just flare the methane or burn the methane as it comes out. But their main purpose of using anaerobic digestion systems is to reduce the odor. Because if you contain all this material within a closed-in vessel, the only way to get that odor out is through a pipe, and if you run that pipe through a flare or some way, you can burn a lot of these bad-smelling odors. You can take care of it. Some of the lagoons I've been through or next to that are anaerobic digesters. One, you can't smell them, and then the effluent wastewater is not anywhere near what the effluent wastewater smells like. So odor reduction is probably one of the top things that you want to look at with anaerobic digestion systems. The next thing there is seasonal treatment. Like I said, we're using fruit and vegetable waste, and the thought is that this time of the year, which is February, March time frame, you may have broccoli coming off in our area. So if we can feed these digesters broccoli this time of the year, and then the next thing that may come off is like cucumbers or squash, and then the next thing and the next thing, and then July watermelons will come off. They'll take a little bit of season break, and then you'll start back up with the fall crop of squash and cucumbers and such as that. So taking care of the materials or the fruits and vegetables that come off during different seasons, you can use that throughout the year. So a seasonal treatment of these materials. You reduce your waste and biomass disposal. So if you can take a lot of these organic materials and put them into an anaerobic digester, for one, you reduce the volume that you have to deal with, because a lot of the material, about 92 to 95% of the material that's in a fruit or vegetable is volatile solid, and a volatile solid is a material that is very digestible by, or is feasible to be digested by bacteria. So it's an organic compound, a carbon-containing compound, and the bacteria can reduce that. So you're looking at, think about buying a bag of potatoes, for instance, or a bag of onions. Out of that 5 pounds, 95% of it or so can be digested by a bacteria. So you only have 5% of that 5 pounds that would be a sludge. So you've greatly reduced your biomass. And then, like I said a minute ago, the anaerobic digestion process actually saves nitrogen, phosphorus, so it flows through the system to be potentially used as a low-grade fertilizer. What I want to do on the next couple slides is look at how the anaerobic digestion system works. Now, I don't want to go through and get too technical, but kind of want to show you the flow of how these anaerobic digesters convert carbohydrates, proteins, and lipids, the fats that's in different materials down into the methane that we do capture and then use. So the first process is this hydrolysis process. It's a breaking of a lot of these long organic compounds, the sugars and carbohydrates, proteins and fats, breaking these materials down into more usable materials. And these more usable materials or smaller materials are those amino acids, as you see there, and sugars. It also breaks it down to these higher fatty acids and alcohols. These may be like C20, C30 compounds, as these higher fatty acids, based on what lipid goes into producing those higher fatty acids. The next step in the process, or this whole process, is the fermentation oxidation stage. And so the next step is those amino acids and sugars go through a process. There you see that number 2, number 3, number 4 process. But it's basically all the anaerobic bacteria are breaking these materials down, cleaving them, making them smaller molecules, and taking those potentially C20, C30 acids and making propionic butyric, the isobutyric, acetic acid. You're looking at C5, C6, C7, C8 type acids at this point, so a lot smaller acids. And then these materials will go down into the next stage, which is methanogenesis process, and that's where these smaller C6, C5, C8 acids are broken down into acetate. And then as you see there on the screen, the acetate and some hydrogen are combined to form that carbon dioxide and methane. And so our 3 basic parts of the anaerobic digestion system is the hydrolysis process, where a lot of those fats, lipids, and proteins are cleaved down into smaller and longer chain carbon compounds. Fermentation, which breaks it down into even smaller acids, and then and specifically acetic acid. Methanogenesis breaks that acetic acid and hydrogen down into methane. What are we looking at when we talk about this methane that we're producing? It's the major component of natural gas, as we all know. It's a colorless, odorless gas. I remember those old commercials, and I guess they still have them out there, that the natural gas lines, they say they put the rotten egg smell in there so you can smell it. It's the same gas that we're looking at here. As you would expect, it's a very explosive gas. And like I said, it breaks down, it's the result of that breakdown of organic compounds in the absence of oxygen. And that's why these tanks that we build them in and put them in are as sealed as they can be. And there's some research out there that shows that if you inject a little bit of oxygen into the system, if there's a lot of sulfur-containing compounds and there's a lot of sulfur being produced, that will actually change the sulfur gas to a sulfur solid so it settles out and you don't have to deal with it. So that there's a little bit of research on that. The symbol we saw up in Goldsberg. And generally one of the reasons we want to look at this is, you can see there's estimates, and I've seen the range of numbers from 20 to 23, but it's this methane traps 20 to 23 times more heat than carbon dioxide. So when we start talking about greenhouse gases, methane is one of those gases that we're really looking at. So based on a lot of that, and when we start looking at this fruit and vegetable waste as a potentially viable source of energy, we come up with these couple objectives that we want to look at, and especially look at during my research projects. And the first area is to measure these chemical physical parameters of fruit and vegetable waste. We started that work a little over a year and a half or so ago. Kind of the initial step of, is there any energy potential in these fruits and vegetable waste? And if there is, then how do we go forward with it? If it's not, then let's don't waste too much time trying to build digesters. And I'll show you a little bit of that data here in just a minute or two. We also want to operate some of these anaerobic digesters on fruit and vegetable waste and kind of really look at what's the biogas production level, what's the yield of biogas per liter of fruit juice input, and we've done a little bit of that with onion, and we're gearing up right now to really start for this year. Look at the feed rates and trying to optimize the feed rates and the solids contents in these fruit and vegetable waste digesters. We want to try to feed them as fast as we can to keep them happy, if you want to call it that, but we don't want to overfeed them and get them obese and basically kill themselves. So it's kind of that fine line, and we want to try to see if we can find that fine line. And then the last objective, like I mentioned a little bit ago, is can we take this fruit and vegetable waste, and if there's a viable source of animal waste or some other ag waste out there where we can co-digest these things together to take care of wastewater, to take care of a waste stream, and then still producing electricity out of it. In Germany, some of the companies over there, they actually use an animal waste as kind of a filler material, but what they'll do is they grow corn, make silage out of it, and then use the silage in the animal waste in an anaerobic digestion system to produce methane. So we'll do something similar to that, except we'll use fruit vegetable waste. When we start talking about measuring these fruit and vegetable waste for chemical and physical parameters, what we're looking at is mainly moisture content, how much water for one, but more how much liquid is in that fruit and vegetable. What's the volatile solids number? And the volatile solids is actually it's a measure or the materials that actually can be destroyed. They're carbon-based compounds that can be destroyed by the bacteria. That COD, again, is just a real quick test that we can do to determine the feasibility and the potential energy in this fruit and vegetable waste. And the bricks, if you do any work with fruit and vegetables, it's just a measure of those suspended solids. And what we're trying to do as well is relate the COD number to a bricks number. The bricks number, to get it, we use a refractometer, and it's a couple second test. The chemical oxygen demand is, like I said, 2.5, 3 hour test. So what we want to do in eventual goals is if we can put some of these digesters at the end of a packing line and run these digesters that way, one of the problems we foresee is that you may be harvesting, for instance, let's say cucumbers in the morning, but that afternoon you may be harvesting bell peppers. So if the digester can take in a kilogram or 100 kilograms of cucumbers, but you need 3 or 4 times the bell peppers. We need to know that, and COD can give us that information. So if we can relate the COD number to the bricks, we can take a quick bricks number coming straight off the line, know that it correlates to a given COD, and then we can dial in, if you want to call it that, the digesters to feed it this COD number. I know there's a little confusing, and maybe I can explain it a little bit more, a little bit further down. What we do, especially here, especially when we start going through this physical chemical analysis, is first we take the samples in, and we take those bell peppers, as you see here, and we process them by cutting them up, and then we'll grind them. And the grinding process will release a lot of that juice. Then we'll send it through a blender, and actually I've purchased now one of the home juicer systems. So we'll take the ground up material or the whole materials, we'll juice it out, and we get our liquid stream. And that liquid stream goes to do these COD measurements. And one thing that I'm looking at specifically is trying to separate the pulp from the liquid. The liquid then we can use in what we call high rate anaerobic digestion systems, and I'll show you a picture of them in just a couple seconds. But we have some kind of matrix in there that the bacteria actually grow on, and you can shoot liquid through there relatively fast, whereas with solids you have to have either an open container or it'll clog up the system on you. So this juice we use We get our COD number, which then leads to how much we can feed the digesters. The other thing we do, we take that ground up bell peppers. They don't magically turn into onions, but I didn't have any bell pepper pictures. But we'll take and also dry that material to find the moisture content, to see kind of what we're looking at from a moisture standpoint. And then that will go into a 550 degrees C oven to get our volatile solids number. And again, that volatile solids is an indication of how much material is there that the bacteria can actually convert over to methane for us. And both the COD number and the volatile solids number are indications of how much we can feed the digesters. This is two of the student workers I had a couple years ago when we were really looking at the different varieties of Vidalia onions here in Georgia. But these two guys, they cut up, I think, over 900 samples of onions over about a 4-week time period, where we took all the variety tests here in Georgia, cut them up, as you see, took a Brix number, and then burned them and ran them through their paces, if you want to call it that. This is some of the numbers that we got from the chemical characteristic test that we did, specifically that COD. As you see here on the far right-hand side, the onions, this number shows a little over 120, and these COD units are measured in what they call grams per liter. So it's a gram of oxygen required, or a gram of COD, oxygen demand per liter of juice. So onion has a little over 120, whereas tomato there you see has a little bit less than about 5. Bell pepper is right around 60. So, for instance, if we're feeding these digesters bell pepper waste on a standard diet, if you want to call it that, if we changed over and started feeding them onion waste, then we'd have to actually feed them half as much onion to keep the same COD going into the system. So knowing these COD numbers for the different fruits and vegetables is needed, because we need to feed the digesters a constant COD number. And again, that's why we want to look at, see if there's a correlation between Brix and COD. Brix we can take real quick, and then if the Brix gets a little bit out of whack, if you want to call it that, or there's a little bit higher COD or a little bit lower COD based on this Brix correlation, then we can speed up or slow down the pumps feeding the digesters. This graph here is just showing some of the volatile solids from our Vidalia onion trials over the past couple of years. We're looking at somewhere between 92 and about 95, 96% volatile solids, which there's a little bit statistically difference between these onion varieties, but generally there's nothing that's going to really upset the apple cart, if you want to call it that, if we put all these different varieties in the same vat, mix them up, and put them through our anaerobic digesters. The digesters we've been using, or I've been using here, especially on the past couple months, are anaerobic filter systems. They are what they call high-rate anaerobic digestion systems. They have a packed media within them. I've been using lava rock. The systems I'm using are roughly 90 liters empty. When I put the lava rock in them, it comes out to be about a working volume of 45 liters. I've insulated them, and we keep them heated up to about 35 degrees C with computer control and whatnot, so that the little bugs inside are pretty happy. And we feed them, we heat them, and then we're looking at how much biogas is produced based on how much liquid we put in, and then everything's controlled and monitored by Campbell Scientific data loggers. This is just a schematic of what these anaerobic digestion systems look like. You can see the little red dots there is the lava rock, and these things operate, and as we flush the water across the lava rock or the wastewater, it's similar to a stone or a pebble or a rock that you have in a creek with the moss grown on top of it. It's that same type biofilm within these digesters. So as the water moves down through the system and over those rocks, the sugars and the proteins and the fats and lipids are exchanged across that biofilm, and as it gets deeper into the biofilm, the sugars and proteins and fats are converted into that acetic acid and methane, and then that effluent is that sludge and liquid water that we can use as a biofertilizer. This is another just schematic of what my system looks like in a cutaway view, except those digesters there they would be 90 liters. The small digester there in the middle would be more like a CSTR system, a completely stirred tank reactor where there's no matrix in there, and we completely stir that tank up. The system I have shown here on this display board is more of a two-stage system, with some of the thoughts that we're going to start running some of these two-stage type systems to look at doing that hydrolysis process in the first stage, and then taking that the short fatty acids, short chain volatile fatty acids, and put them into that second stage digester or anaerobic filter to get a lot of our methane out. You can see a picture of what our data logger looks like, and this picture here on the right hand side there above the second digester is just a tipping bucket. It's an upside down rain gauge is what it is, and we measure gas flow with that. Is just a picture of the 3 different digesters, or a set of digesters. These are my 90-liter lab scale digesters. You can see the blanket around them to keep them warm. And what I have under the blanket is a series of pipes that actually is like a water blanket that keeps them warm. These 3 digesters are running in parallel with each other. I feed them onion juice, or these have been fed onion juice, and I use the 3 different digesters as replications of each other. The white box there contains our computer, the CR3000, and then we feed them. You can see the lines coming in. So I can find my arrow here. You see the feed lines coming here where we pump the liquid wastewater in from the room next to this one. This is just a closer view of what our tipping bucket looks like. Like I said, it's just an upside down rain gauge, and as the gas would come in at this point where my arrow is, it would flow up as the arrow is flowing, and then this has here at the top just a magnet. As those magnets contact each other, it counts a pulse, electrical signal, and that's recorded in our data logger. So right now what we're doing is we're taking onion culls mainly, or we're taking the onion culls and we're separating the fruit from the pulp from the juice, and we're running those juices through those anaerobic digesters I showed you a second or two ago. We started out feeding them 800 or 500 milliliters every 8 hours, and we continually increase that to 1,000, 1,500 milliliters every 8 hours. Somewhere between 1,000 and 1,500 milliliters, it got too much sugars going into the system. It really crashed our systems. The pH dropped down to about 2.5 or a little bit lower, killing the systems. And even today, after running dairy waste through these digesters for about 3, 4, 5 months now, you can still take a sample out the end of them and smell onion juice in them. So these things, like I said earlier, they're pretty stable if you feed them the same thing day after day after day. But when you start excuse me, tinkering with them and trying to increase it and such as that, sometimes they don't like it, and they will crash on you. Temperature, again, is controlled by the computer. This is just one of the graphs that we noticed from the biogas production. We were feeding these digesters, in this case, every 12 hours. Well, we would feed them at midnight and noon every day. And as you see, it would take about 3 hours from the time of feeding to get our maximum gas output. And then within 3 or so hours after that, we'd almost get no gas production whatsoever until our next feeding rate. What we tried to do, as you see here in this slide, is we tried to close down um, close down a little bit on that time, but also increase their feed rate. So like I said a minute ago, we went from 500 milliliters up to a liter and then up to 1,500 milliliters. And then once we got to that 1,500 here on the right-hand side of our graph, at this point, the digesters just got too much acid in them, and it crashed the system. All those acids just were too much, and it crashed the system, dropped the pH below. But this is when we start the digesters again here in a couple of weeks. This is what we want to try to mimic and start looking at this go around. How do we co-digest with the animal waste a little bit better from a 25-75 split of onion juice to animal waste to a 75 onion juice to 25 animal waste. So in summary, what we've been looking at mainly is anaerobic digestion using these fruit and vegetable wastes, for one, to get rid of the potential source of water pollution. Use this fruit and vegetable waste, try to capture some of that solar energy that came in, and then use the liquid and the sludge fraction as a soil amendment. And then we're looking at, trying to find the best methane yield per liter of juice input. And then so as we are starting to look at this fruit and vegetable waste, we kind of replace the cows on that cartoon we saw earlier. And when we start looking at the green energy, I'm thinking more of green bell peppers more so than other green sources. And Vidalia onion is Georgia's state vegetable. So if we can use the state vegetable to produce energy, I think we may be actually a little bit ahead of the game. Just some acknowledgements. Most of my funding comes from the Department of Natural Resources Sustainability Division that funds our program. We've got a USDA grant last year through the Specialty Crops Research Program, and then we also have some CIG grant funds from NRCS that help fund this research that we've been doing, both education and research. And with that, Mike, I guess I'll turn it back over to you or see if there's any questions out there.
Thanks, Gary. We do have some questions that have come in. One came in from Osa. I don't know if he's on a microphone. I will try to unmute you. Osa, can you? Are you on a microphone? Okay. Well, I'll just read his question. He asked, "Is the quality of the biogas affected because you're not using rumen animal manure? Does that change the quality of the biogas?"
It doesn't seem to be so far, and it shouldn't be. Biogas should be biogas coming out. We just get it a little bit quicker. So no, it shouldn't change that at all. John, did you have a question? I saw your hand came up, but then it went away. John, did you have a question? Chuck, what's your question, young man? Are you out there, Chuck?
Oh, Gary, can you hear me?
Yes, sir.
This is Chuck with Sustainability Division.
Yes, sir. What's your question, Mr. Chuck?
I was wondering, at your choice of chicken litter for your carbon source, you're also throwing a lot of nitrogenous material in there, and I was wondering if you tried sampling with, say, using sawdust or maybe 1/4 1/8 minus wood chips without the additional nitrogen, or a pure source of carbon.
So far, we haven't, Chuck, but some of the things that we do want to look at here coming up pretty soon being the next year or so, is using some of those other materials like you mentioned, sawdust, maybe even some yard waste. I know that has a little bit more nitrogen in it than probably what we would want, but we've looked at some of that. Some of the stuff I've also started looking at is down here in South Georgia, we have a lot of peanut shells. And so we've even started looking at replacing the the lava rock and our anaerobic digesters with peanut shells and seeing what's the degradation rate of the peanut shells so that we could feed them in as the matrix and the bacterial carrier. So we're starting to look at that, but we haven't got too far down the road on that.
That sounds like a real promising idea. You deal with another waste at the same time.
That's kind of the hopes and dreams, if you want to call it that, and I'm trying to find a grad student now to kind of take those projects on.
All right.
Any other questions?
Gary, we have a question from Osa. Let's see if he's on the line now. Osa, can you are you on a microphone? Still not there, apparently. I did see his hand go up.
Yeah, I see it too.
Okay. We see. We did have another couple of questions here. One is again from Clay Olson, and that is, what's the progress on the renewable portfolio standard for the state of Georgia?
Ooh, I hate to say, but I have no idea. I hadn't really kept up with that too close. Chuck may be able to answer it better than I, but right now, as far as I know, it's still the same as it was when we were up in Goldsburg. That hadn't made much of a progress.
Okay, and I see Osa did type in his question, another question, which is, if I can read it correctly, in a real-life case...
Hold on a second, Chuck. Hey,
yeah.
Mike, hold on just a second.
Okay.
Let me bring Chuck back on the line here for a second. Chuck, did you have any...
To the best of my knowledge, we do not have an RPS in the state, and I am not aware of any legislation that has been introduced in the current session of the state legislature. So it's probably going to be a moot point for at least another year, unfortunately.
Okay, thanks, Chuck.
Yes, and Osa's question was, in a real-life case, if a farmer wanted to go into this, what are the issues that you see with multiple produce and vegetable products or inputs as far as control goes?
I think there's a couple different issues that come into play. If you're looking at a single producer producing multiple fruits and vegetables, it comes back to that COD. Let me see if I can find it real quick. That COD graph that I showed you here, that should be on your screen now. What you have to do with these digesters is you have to feed them the same thing day in and day out. So the old analogy is, if you're going to feed them hamburgers every day, you feed them hamburgers every day. You don't throw chicken breast in there, or you don't throw a steak in there. You feed them the same thing. So one of the things we want to do, and the way we feed them, is this COD number. Or we can feed them volatile solids. It's a little bit harder number to get. It's more of a 2-day process than a 1-day or 2-hour process. But if we want to feed them, for instance, on this graph, if we want to feed them watermelon juice every day, that's fine. They can take it with some control. They can eat watermelon juice every day. Now, if you go in there and you throw in onion juice on top of them, it's overloading their system too much, and the acetic or acid forming bacteria will really ramp up the acid production, and the methane bacteria that converts that acid to methane can't handle the additional acid, and it drops the pH and kills them. So that's one reason we want to look at this COD number and try to take that Brix number that I mentioned and correlate the two, so that if we go into a situation where we're feeding them watermelon juice and then we start ramping up, let's say, bell pepper production, and we have a lot of bell pepper juice, if we can take that quick Brix number and back off our pumps a little bit so that we don't feed them as much COD into the system, then the digester should go. So I guess to answer your question, as long as we feed them a constant stream of COD, the vegetables should not matter. I hope that answered the question. Any other questions out there?
Gary, yes, we have a question from Lindahl Harned over in Kentucky.
Okay.
His question is, you were, and this again came in typed in. You were feeding the digester every 12 hours and getting energy spikes. Have you tried a slow drip or a constant feed of juice to maintain a constant energy production?
Linda, we haven't yet, but that is our next step, is to try to back off that a little bit and do drip it in there a little bit at a time, or try to reduce the COD input but increase the frequency. So yeah, we're in that process right now to try to get those spikes down. Any other questions, Mike, that came in over the chat box there?
Yes, here's another one from OSA. How do you measure the H2S? How many ppm does the biogas have from your production?
Also, I have not measured the hydrogen sulfide content, but just smelling the gas output, and I know that's not the most technical way of doing it, I don't seem to have much sulfur in the system, especially from these fruit and vegetables, but we hadn't measured that sulfur content, either H2S or any other form in our digesters yet. But it doesn't seem to be a problem, at least from an odor standpoint.
Okay, I'm not sure I'm getting these in order, but Luis Rivera is asking, what is the use of the biogas?
Luis, right now we are just mainly looking at yield of biogas, but there's a couple different uses that can be done if you're producing enough gas. One of the onion operations, Gill's Onions out in California, are actually running their biogas or their methane through fuel cells to produce electricity. Some of the guys in Vermont are using that gas and running it through generators. We have one, the only anaerobic digester we know of here in Georgia is running that biogas through a modified diesel engine. And then that energy, electricity is going back on the grid, and also to run his operation. A lot of guys will use just the methane gas itself to run a boiler and produce either steam or heat. So there's a couple of different uses. One thing we also want to look at with some of our onion waste is, can we compress that a little bit and put it in fuel tanks, and then run forklifts within buildings off of that methane gas. So there's a couple of different uses of it, but so far we are just in that trying to figure out how much gas per liter we can get out of these fruit and vegetable waste. But eventually, those are some of the uses of that gas.
And Gary, we had a question from John Ignash quite a while ago. He said, is the anaerobic digestion on the juice only, or can the blended solids be digested too? In either case, what becomes of the solids?
John, we can use either or both. With that, let's see if I can find it here on my presentation. I can't find it right now off the top of my head. But in that dual system, like you see now, hopefully on the screen, and what we're doing at my lab, I've got a 1,500 gallon, what they call an ASBR system, an anaerobic sequencing batch reactor. It's similar to a CSTR system. Like I mentioned, there's no matrix system within the digester. So in that type digester that doesn't have a matrix in it, you can put both solids and liquids in the digester. The solids will go through that breakdown process just like the liquid fraction will. The only thing is, with the high rate systems, with the lava rock or some other matrix in it, you can't put the solids through as you would expect. It would clog the system. But if you have a digester that's designed and built to handle solids, it can take the solids. Generally, the high rate filter system or the high rate systems with a matrix in it, you can potentially get what they call hydraulic retention times. You can shoot the juice through it in a matter of days. Generally, when you expand that to take solids in, you're looking at more of a week or 2 weeks type. So your time frame to break that material down expands a little bit, but that kind of makes sense because you've got the solids there. I hope that answered that question, John.
Okay, Gary, we have Leif Kinberg has put his hand up. Let's see if Leif is on a microphone.
I'm here.
Hey, Leif.
Gary, very interesting presentation you're giving.
Well, thanks, sir.
Do you have a sense, from the economic side of it, do you have a sense of both scale in terms of power generation and cost, basically the cost that would be required to make these economical at some smaller scales?
That's kind of a tough question, Leif. And we're starting to do some of those economics on these digesters that I have here, especially that 1,500-gallon tank system. Generally, it really depends on what you want to do with the biogas as an output as to what the economics are going to be on the input side, if that makes sense. If you're a small producer, and this is not really based in research because we hadn't, or at least what I've done and seen, but the smaller systems, if you want to use that material for heat or small generation, you're okay. But larger generation, obviously, you're looking at millions and millions of dollars to put these systems in, but you've got to have the feedstock there to handle that that side of it. But what we're trying to do is look at what's the smallest system available that a small producer can use on farm to take care of their small operations. I know that was kind of a vague answer, Leif, but we're still kind of in that phase of what's that break point to make it a viable system economically. And two, that's one reason we're starting to look at or trying to look at these high rate systems where the capital input is a lot less because, for instance, if you're using a completely stirred tank reactor, excuse me, you have to, generally speaking, you design those for a 20-day retention time to keep the liquid and or solids in there for 20 days. These high-rate anaerobic digestion systems, you're looking at 1 to 2 days. So the capital input or the size of the vessel can be sometimes 20 times smaller, 10 to 20 times smaller. So the capital input for those systems are going to be, at least 10 to 20 times smaller. So, but right now, like I said, me and one of the economists here are taking the systems that I'm building at my lab and really going through a good economic analysis of it. So to tell you it's going to be viable at this level, I can't do that right now, Leif.
Thanks, Gary.
Okay, Gary, we'll take one more and then maybe we'll wrap it up. Again, this is coming from Osa. He wants to know what kind of support you have for grad students who want to work with you and at what level.
Right now, we've got a couple grants that have grad students built into them, either at both the master's and PhD level. And we're in the process of drafting up a couple more proposals, hopefully, that will get funded, that will have graduate student monies or stipends written into them as well. So right now we've got one to look at either a master's or PhD student. And then, like I said, we're in the process of drafting some more.
Okay, well, thank you. And I would certainly encourage everyone to follow up directly with Gary if you have more questions. And would like to thank Gary very much for his excellent presentation. Thanks to you all for your participation and for your good questions. This concludes today's webinar on behalf of the Energy Training for Ag Professionals program. Again, thanks for participating, and have a good day, everyone.
Thanks, everybody.